Welcome to part 2 of lecture 4. We are going to be talking about the different types of transport in this video. So, one of the easiest type of transport to understand is the concept of diffusion. Okay? Diffusion involves the movement of molecules from an area of high concentration to an area of low concentration. So the best way to visualize this is think about what happens when you add a drop of food coloring into a flask of water. Okay? So when you add the dye is very concentrated in one area, and over time you will be able to see that the dye will diffuse outward, and ultimately when given sufficient amount of time, the entire flask is going to turn blue. You don't have to do anything for this process to occur. Once you add the dye, it's only a matter of time before the whole thing is going to be blue. It's a natural process that will occur, and what happens is molecules tend to move down their concentration gradient spontaneously. A concentration gradient basically describes a difference in concentration between two areas. So over here you have a lot of dye in a small area, so this is high concentration, and over here there's no dye, so there's a low concentration. And naturally molecules will move from an area of high concentration to an area of low concentration, and this is going to happen until the whole flask is evenly distributed with dyes. Another example of this would be, say you open a bottle of perfume in one corner of the room, and soon you will be able to smell it on the other side of the room, right? That's because the scent in the perfume bottle is very concentrated, and once you open the bottle, the rest of the room has no perfume molecule, and therefore it's considered as a low concentration. So things will spontaneously move from high concentration to an area of low concentration. This concept is called diffusion. Now, there are different factors that are going to be affecting the rate of diffusion. That is, how fast will diffusion take place? The first factor is the size of the molecules. Okay, so something very small, like this O2 molecule, they tend to diffuse a little bit faster than the larger molecules, like glucose shown here. And if you have something like a macromolecules, then they will be super slow compared to the diffusion of something like oxygen. But size alone is not the only factor that determines the rate of diffusion. Temperature also plays a role. Typically, if you have higher temperature, it would allow things to move a little bit faster, and therefore it will result in faster diffusion. Also, the steepness of the concentration gradient is also going to determine how fast the diffusion will occur. So, for example, in this picture here on the left, you have a very concentrated area. Okay, so this is high concentration. And I'm just gonna put a number here to for illustration purposes, because it's filled with these things. Let's say this is 80% concentrated. Okay, 80% concentrated. And over here on the right side, this is low concentration and it has absolutely no red dots, so it's 0% concentrated. So comparing 80% to 0%, then the difference is going to be 80 minus 0. So we have a difference of 80%, right? Triangle means difference. So that's how steep this concentration gradient is. Over here, right, on the left side, we have some red balls, and let's just say this is 70%, okay? And it's still considered as the high concentration compared to the right side, which only has a few balls, so this is low concentration. But it's not 0% over here, right? There is still, because there's some red dots here, and so this might be, let's say, 10%, okay? So if you do 70 minus 10, the difference here is 60%. So because there's a greater difference here between the two areas, then the concentration gradient is steeper in this case than it is over here. So diffusion is going to be expected to go a little bit faster on the left side here compared to the right side because it's steeper. Okay, so the greater the difference between the concentration between 2 points, the faster the rate of diffusion will be when the molecule moves from one place to another. Finally, there is the effects of charge on ray of diffusion. If things are neutral, meaning it's not positively charged or negatively charged, then the other three factors would affect the ray of diffusion primarily. But if it's charged, then the difference in charge between two regions is going to affect how fast diffusion is going to occur. So let's say we have a bunch of positively charged hydrogen, right? We call these protons, as you might remember from earlier lectures. So here we have an area of high concentration for the protons and low concentration of protons. And what happens is, because they are all positively charged and like charges, they repel each other. So not only will the H plus diffuse down its concentration gradient, but because of the electrostatic repulsion, the like charges repelling themselves, that's going to give it an extra push for these hydrogen to go over. Okay. Now there is something else that's worth mentioning here, and let me just try to exit this presentation view to show you. Initially you will see these things go really fast over to the other side, because like I said, the positive charge will repel each other, right? However, as the molecules go over, it's going to have a harder time moving forward down the concentration gradient, because now there is a buildup of positive charge on the right side here as well, right? So this thing, if it wants to go over, it's not going to be as easy, because all these positive will kind of be pushing this back as well, right? So there's going to be a point where the diffusion rate is going to be counterbalanced by the charge repulsion, and even though you have a really quick diffusion at the beginning because of the buildup of charge, later on that buildup of charge on the other side will actually slow down the diffusion. Okay, so this one is a little bit more complicated to understand, but do think about it. And you know, play around with these diagrams to see how it affects the ray of diffusion. The next concept is called osmosis. Osmosis is basically the diffusion of water across a selectively permeable membrane, such as a cell membrane. So let's take a look at this diagram. Okay, here we have a U-shaped tube, and it's filled with water, but separated by a selectively permeable membrane. So this membrane will only allow water to move, so the blue stuff to move across it. The red stuff will not be able to go through. Okay, so just to refresh your memory, a solution is going to be made up of two things, right? There is going to be the solvent, in this case is water, and then there's going to be the solute. Okay, the solute would be the red dots in this case. Okay, so probably I should write solvent, and this would be the H2O, which is shown in blue, and then we have the solute, which is the dots. Okay, so on the right side here we have a high concentration of the red dots, and on the left side we have low concentration of red dots. Now, if this membrane were not selectively permeable, if this membrane allows everything to go through, then the red dots would go from an area of high concentration to an area of low concentration, right? So again, if this membrane lets everything through, then the red dots are just going to move over. Okay, so it's a little bit more complicated in this situation because of the selectively permeable membrane here. This membrane does not allow the red dots to go through it. So even though these red dots, which are really concentrated on the right side, wants to go to the left, they cannot physically pass through this membrane because they're too big. So to balance the concentration between the two sides, the only thing that can go through would be the water molecules. So here we have high concentration of solute, and here we have low concentration of solute, and it's only water that can pass through the membrane. So the water will actually shift to the right to dilute out the red dots on the right side, so that the left side and the right side will end up having the same concentration. So over time, what you will see is the water level will rise on the right side, and then the water level will go down on the left side. Okay, so you might want to listen to that explanation one more time. It's a little bit confusing because we are so used to thinking things go from high concentration to low concentration, right? But the problem here is this membrane will not allow the red dots to move, and so only water can move. Okay, and whenever this situation arises, water will always move from the area of low concentration to an area of high concentration, and this kind of movement of water is called osmosis. So an example of this happening in real life would be, say, preserving your food by adding a lot of salt to them, right? So if you put a fish in a bunch of salt, or your meat or whatever, then the salt is actually going to suck the water out of the meat, right? Because the salts are too big to physically enter the meat, and so here the skin here serve as like a semi selectively permeable membrane, and so when you have a lot of salt on the outside, the water will be sucked out of the meat, moving from an area of low concentration inside the meat to an area of high concentration, the salty environment, basically. Okay, and by removing a lot of the moisture from the meat, you're able to prevent bacteria growth, and therefore preserve your meat for a very, very long time. So now we are going to look at the concept of water balance in a cell, and we are going to take some class notes to explain this concept. So I encourage you to draw this along with me so that you will understand what is going on. Here is a typical animal cell, okay, animal cell, and we are going to be placing the animal cell in different environment, and we're going to look at what happens to each one of them. Okay, so in the animal cell, there will be various dissolved ions, electrolytes, and proteins and whatnot, and all those things are going to contribute to the concentration of the fluid inside the cell. Okay, in other words, the cytoplasm is going to have a certain concentration associated with it. In the best setting, that concentration is going to match the concentration of the cell's environment. Okay, so if this is going to be I'm going to give you some number. Let's say this is 70% concentrated on the inside, and the outside is also 70% concentrated. Then what's going to happen is the environment will match completely with the inside of the cell. So the amount of water that's moving out will be equal to the amount of water coming in. Okay, and in that case, the cell will neither be gaining fluid or losing fluid, and it's going to be quite happy. These kind of cellular environment, we call it isotonic. Okay, isotonic. Right. So there's no net gain or net loss of water. The rate at which water exits the cell is same as the rate at which water is coming in. So the cell will remain the same. Okay. Now we're gonna look at a second scenario. Okay, so maybe I should put here no change in cell volume. Okay, over here we have another animal cell. Let's keep it as 70% here, and I'm gonna put some pink dots here for you. So same concentration of pink dots as before, but now we are going to be putting this in an environment where there is not a lot of pink dots on the outside. Okay, so let me just separate these two cases with a line so we don't get confused. So now we have more pink dots on the inside than we do outside. And again, right, you might be thinking, well, then the pink dots are gonna leave the cell, right? But not quite, because the cell membrane is selectively permeable, right? So it might not let the pink dot leaves the cell. So the only thing that can move in this case would be the water, right? And water will always move towards the side that has a higher solute concentration. Think about salting the fish, right? It will move towards the side with more salt. Okay, so here we have more solute, right? More salt, so to speak, inside the cell. So you will have more water coming in. Okay, so you have a lot of water coming in. It doesn't mean you don't have water going out. You will still do, but the key thing here is there are more water coming into the cell than water that's leaving the cell. And so over time, what's going to happen is the cell is going to swell up, and it's going to get really big. Right? It's going to fill up with water, and eventually, what's going to happen is if this is an animal cell, then it might explode. Okay, so explode. Okay, all the stuff is going to leak out, and then the cell is basically going to die. Okay, so this kind of environment is called hypotonic. Okay, So hypotonic, you can remember it. The cell gets really big in a hypotonic environment. Okay, and it's gonna get so big that it's gonna burst, and that's going to kill the cell. Okay, so this is called lysis. Okay, lysis is to break. There was once a competition called Hold Your Wii for a Wii. Okay, Hold Your Wii for a Wii. Right. So some of you might know that the Wii is like the Nintendo gaming system, and when it first came out, they held a contest where contestant has to drink as much as water as they could and hold it in their body. So one of the contestants who won the contest actually died shortly after the competition because all that water she drank caused her bodily fluid to be very diluted. So it caused her cells to be in a hypotonic environment, and so a lot of her red cells are basically burst, and that resulted in an unfortunate death. So the next scenario that we're going to look at is what's called the hypertonic solution, hypertonic environment, right? And as you can guess, it's going to be the opposite of what's happening here. Okay, so let's draw our regular cell, and we are going to have 70% concentration here. Here, just to backtrack a little bit, right? I forgot to put a number on the outside, but the number outside would be lower than 70, right? Let's say I put 10% there, right? So over here, we have the hypertonic solution environment. Okay, the inside is gonna have these pink dots just like before, and then the outside will also have the pink dots, but then you would have a lot more. Okay, so you will have a lot more. Okay, so if I were to put a number here for this environment, that might be, let's say, 95%. Okay, so again, right, you might think the pink dot's going to come in from high to low, but you can't because of the selectively permeable membrane. So the only thing that can move is water, and water always move towards the side that has more concentration. Okay, the side that has more salt, right, so to speak. So you would have a lot of water leaving the cell. Okay, that's not to say water will not come in. They will still come in, but the key thing here is you have a lot more water leaving the cell than you have coming in, and so you're gonna have a net loss of water. Okay, so the cell is gonna shrivel up like this. Okay, and this is going to be called plasmolysis. Plasmolysis. Okay, so the cell will still live. Okay, in fact, you have special cells in the brain region called the hypothalamus, and anytime you are dehydrated, right, your blood concentration goes up, and it will cause those cells in the hypothalamus to shrink, and that shrinking will actually send a signal throughout your brain and tells you that you are thirsty, and so you will drink some water, and by drinking the water, you will hopefully bring it back to the isotonic environment. Okay, so that's 3 examples of what happens in an animal cells. Let me just show you what they look like in the PowerPoint here. You can see in the isotonic environment, right, the cell is quite happy. This is a red blood cell. Amount of fluid going in and out cancels each other, so the cells don't gain or lose any volume. In a hypotonic environment, the water coming in is going to be much quicker than the water is leaving, so the cell will swell up, and it will result in lysis, or the bursting of the cell. And on the other hand, if you have a cell that's placed in a hypertonic environment, then the water will be leaving to the outside, and your cell is going to shrivel up. Okay, it's probably worth mentioning here, if this was a plant cell, then it's going to be a little bit different. Okay, plant cells actually like the hypotonic environment because they need to gather a lot of water for photosynthesis. So plant cells will be in a hypotonic environment a lot of the times, but they're not going to burst because they have the additional cell wall, right? So the cell wall will help protect the cell. Okay, think about like inflating a balloon. Okay, if you inflate a balloon, keep on putting air in, it's eventually gonna pop. Okay, so that's kind of like the animal cell in the hypotonic environment. But for the plant cell, it's like putting that balloon in a glass in a cup first, and then you inflate the balloon. Okay, so the cup is going to serve as an extra protective barrier to prevent the balloon from bursting, right? Okay, and that's what the cell wall is. The cell wall is the cup that will help protect the plant cells from bursting. Let's take a moment and look at IV solutions. IV solutions are intravenous solutions that are going to be given to the patient through a needle, right? And these IV solutions are actually tailored to the conditions of a patient. Okay, so for healthy patients, the IV solution is of course isotonic to the cells. Okay, you don't want to be giving fluid to someone that would cause their cells to swell up and potentially burst, or to cause the cells to shrivel up. Okay, you want to give them isotonic solution where there's no net movement of water. And so these isotonic solutions are typically 0.9% normal saline. Okay, saline means salt, right? NaCl, and it's written as one of these abbreviation. It's one of the most common isotonic solution that is given for people who need rehydration. So if someone is bleeding a lot and hemorrhaging, right, because they came from a car accident or something, or if they have been vomiting a lot and experiencing diarrhea, you want to replenish the fluid as quickly as possible, and then you will give them normal saline. Okay, so you will rehydrate them without causing their cells to burst. On the other hand, we have 0.45, which is half the concentration of the normal saline, and this would be considered as hypotonic solution. Okay, so you would want to have a hypertonic solutions for patients who are experiencing hypernatremia. Okay, so hypernatremia is too much sodium in the blood. Okay, so if they have too much sodium in the blood, you need to dilute the blood, right? And giving this half normal saline will help you achieve just that. Okay, because if you don't dilute the blood, then the cell might be shrinking, right? So we just want to re-equilibrate it back to isotonic. Diabetic patients sometimes can experience what's called ketoacidosis. There are some chemical called ketone in diabetic patients as a result of metabolism from lipids, and too much of that can cause your blood to become acidic. And by diluting the blood with half normal saline, again, you help it bring back to the isotonic state. Lastly, we have 3% hypertonic saline, and as the name suggests, this is going to have higher salt concentration than within the cell, and you would give this to someone who are experiencing hyponatremia. Okay, so for example, that contestant that I was talking about, who died from the drinking too much water in the contest, if she arrived at the hospital quick enough, they might be able to save her by giving her 3% hypertonic saline, right? Because her blood is too diluted from all those water that she drank, right? So by boosting the sodium content in her blood and giving her this kind of IV might just save her life. Okay, so you probably will learn about the different types of IV solution if you plan to study in nursing, for example. But this is just an overview of some of the common solutions out there. So back to transport, there are basically two categories of transport. We have active transport and we have passive transport, and then there are examples of each type that we're going to look at. So the main difference between active transport and passive transports are summarized in this table. For passive transport, you will not be requiring energy from the cell, so no ATP is needed, whereas you will need ATP for active transport. Passive transport will involve movement of molecules down their concentration gradient, so from an area of high concentration to an area of low concentration, such as diffusion. On the other hand, for active transport, you are moving it in the other direction, going from low concentration to high concentration. So passive transport is going to be relatively easy because it doesn't require energy expenditure from the cell's perspective. It's like pushing a rock down the hill. It's just going to go. Whereas active transport, it's a little bit tougher. You have to push the ball up this hill against the gradient. So in a similar manner, the cell will have to use ATP, use energy to transport things from a low area of low concentration to an area of high concentration. So now let's take a look at passive transport and some examples in that. So the first example of passive transport is something we already learned, simple diffusion. Small molecules like water, hydrophobic molecules such as steroid hormones, as well as some gas, they will have no trouble crossing the cell membrane on their own by simple diffusion. If it's small enough, it's able to fit through the tiny gaps between the phospholipid bilayer, and if it's hydrophobic, then it has no problem crossing the hydrophobic tails layer that are kind of in between the phospholipids, right? You might remember that's what the cell membrane looks like. There's a thick hydrophobic layer in between. So if a molecule is hydrophilic, it's polar, it's going to have a hard time crossing through it because of this big lipid barrier in between. But if it's a hydrophobic molecule, then it's just going to go right through. Okay, so simple diffusion does not require energy, and the molecule will simply go from area of high concentration to an area of low concentration. Now it's worth mentioning that the simple diffusion does not really tell you which way the molecule is going to move, right? So if I have the cell here and I have a lot of O2, for example, on the inside, and then not a lot of O2 on the outside, then the oxygen is going to go out. Okay, but if the condition flipped, if there are more O2 outside than the inside, then now the oxygen is going to be coming in. Right. So simple diffusion doesn't mean that the molecules will always be going into the cell, or is always going to be going out of the cell. It all depends on which side of the cell has a higher concentration. Okay. Osmosis is another example of passive transport. This is the diffusion of water, and water will move differently depending on whether the cell is placed in an isotonic, hypotonic, or hypertonic environment. So for bigger molecules and charged particles, if they want to cross the cell membrane, then they have to use something called facilitated diffusion. In facilitated diffusion, you will use a transport protein, and this transport protein kind of provides a way for these bigger molecules or charged molecules to pass through the cell membrane without interacting with the hydrophobic layers that is in the cell membrane. So in these cases, the molecules are still moving down their concentration gradient, and therefore no energy input is required. So there are two types of facilitated diffusion. There could be a channel protein. Okay, so a channel protein is something like this. It involves an integral protein that spans the entire phospholipid bilayer, and in the middle of it, there is going to be a tunnel. So the molecules would just go through these special tunnels, and they would go into the cell or out of the cell, depending on which way the concentration gradient is. Okay, so large molecules and ions will be able to use this as a way to pass through. Alternatively, there is something called a carrier protein. Okay, the carrier proteins are kind of like a revolving door or the turnstile at a subway station. The molecule is going to bind to the carrier protein, and that's going to cause the membrane protein to change shape, and in the process of changing the shape, it will translocate, it will move the molecule to the other side. Okay, so just think of the revolving door, right? You go inside at one end, you push the revolving door, so the door is kind of changing shape in a sense, and then you end up on the other side. Whereas the channel protein, there's no change in shape for the membrane protein. It just provides like a tunnel for you to go straight through. Okay, so both channel protein and carrier proteins are examples of facilitated diffusion. Okay, and something like glucose would be going through the cell membrane by using a carrier protein. So there are factors that affects the permeability of the membrane. Okay, typically speaking, only small hydrophobic molecules will be able to pass through by simple diffusions, and typically you will have charged particle and glucose going through these channel proteins, but those conditions can change. Certain conditions will cause the membrane to be less permeable, and in other cases it will cause it to be more permeable. For example, if you change the membrane composition, that's going to affect it, right? Say we put more cholesterol into the cell membrane, then now it's going to be very fluid, so things that otherwise wouldn't be able to cross the cell membrane on their own might now be able to do so. Okay. On the other hand, if you remove cholesterol from the cell membrane, then it might become too solid. So things that might normally be able to go through will now not be able to do so. Temperature will definitely affect it. If you increase the temperature, then the cell membrane tends to be more fluid, and then it's going to be more leaky. Changing the pH, changing the ion concentration will also affect the membrane permeability, depending on the kind of cell membrane we're talking about, right? So different cells have different cell membrane composition. I mean, they're made up of the same stuff, okay? They all have phospholipids and you know, proteins and all that stuff, but the percentage of these things are going to be different, and depending on the percentage of the components, the pH and the ion concentration will affect them differently. Also, by changing whether the environment is hydrophilic or this should say hydrophobic rather, hydrophobic. Okay, whether the environment is hydrophilic or hydrophobic, that will also affect how molecules will cross the cell membrane. So you will actually be doing an experiment. Well, since we are doing an online delivery, it will be a virtual experiment on beetroots. We will be looking at how temperature will affect the permeability of beetroot cells. Okay? And we'll be able to tell by looking at how much pigment is going to be released from these cells when you subject them to different temperatures. Okay, so more on that later. So that's it for passive transport. Now we're going to look at active transport. Okay, and just to reiterate, active transport requires ATP, and you will be moving things from an area of low concentration to an area of high concentration. So just to quickly recap, right, we talked about the different types of passive transport. There's simple diffusion, where small things like water and gas can pass through the membrane without assistance. And then there's facilitated diffusion that involves, say, carrier proteins for big molecules like glucose. And then there are channel proteins that provides a tunnel for charged particles to go through. So now we will look at the active transport. In active transport, one example would be what's called a protein pump. Right? So the protein pump involves a membrane protein that will use ATP to pump various things against its a concentration gradient. An example of an important protein pump is something called the sodium-potassium pump. Right? The sodium-potassium pump is important for maintaining the normal condition for your neurons, your brain cells, to function properly. In order for your neurons to communicate with each other, it needs to have a low concentration of sodium on the inside. So let's say this is your nerve cell. The inside must have low sodium, and it must have high potassium on the inside. Okay, so that means the cell must constantly be removing sodium from the inside, and that would cause the outside to have high sodium, and the cell would keep on bringing in potassium, which would cause the outside to have low potassium. And so if you want to transport sodium out of the cell and you want to bring potassium into the cell, this actually is going against their concentration gradient. Right? You can see sodium is moving from an area of low concentration to high, and that's the same case for potassium. That's why this is considered as an example of active transport. So the sodium potassium pump is shown here. It actually has 3 binding slots for sodium and 2 binding spots for potassium. And each cycle that the pump works, it's going to be bringing in 2 potassium in exchange for 3 sodium out of the cell. Okay, and don't worry too much about this glucose things over here. We are just going to be focusing on the sodium potassium pump, and in order for you to do this, you will have to extract the energy from the ATP, and so the ATP will be converted into ADP and phosphate, and the energy will be used to transport the sodium and potassium against their concentration gradient. Okay, maybe I'll just briefly talk about this. Right, so sometimes you can use this concentration gradient that you've created, okay, because you're actively pumping sodium out of the cell. So now there's going to be high sodium on the outside and low sodium on the inside. So the sodium really wants to come back in. Right, it's kind of like creating a dam. Okay, you are creating a reservoir of sodium, and they really want to come back in. Right, and so you provide another channel protein. This is not related to the sodium potassium pump. You have a separate channel, and what happens is as the sodium is coming in, you can harness that energy to transport glucose against their concentration gradient. Okay, so let me just explain that one more time. Here you can see there is high sodium, right? High sodium, and on the inside there is low sodium. And the reason you have this is because of the sodium potassium pump, right? You keep on pumping sodium out, so the sodium really wants to come back in. Over here we have glucose on the outside, and that's low glucose, and on the inside you have high glucose. Okay, so glucose is a resource that we want to keep in the cell, right? So if you want to bring more glucose into the cell, you would need energy. Okay, so rather than using ATP to bring glucose in, right, or rather than waiting for a facilitated diffusion to happen, if you want to actively bring the glucose in, then you can harness the energy from the sodium concentration gradient. So as the sodium comes back in down its concentration gradient, you can use the energy to pump glucose from low concentration to high concentration. Okay, this is equivalent of putting a turbine in a waterfall. As the water falls from a high area to low area, you'll be able to turn the turbine and create energy, and you can use that energy for something else. In this case, you're using the energy to transport glucose into the cell. So there is the protein pump. An example of that is the sodium potassium pump. The last example of active transport that we will be talking about will be something called bulk transport. Okay, and as the name implies, bulk means a lot and sometimes really big. Okay, so bulky material such as an entire bacterium or large particles such as cholesterol, if they want to move across the plasma membrane, and if you want to move a lot of them at the same time, then you are going to be doing something called endocytosis or exocytosis. So in endocytosis, you are bringing it into the cell, whereas in exocytosis, you're going to be expelling it out of the cell. The bulk transport is also used when the cell needs to move large quantity of material into and out of the cell. Okay, so not just bulky stuff, but like if you want to move a lot of them at the same time, you will also be using the bulk transport. So here is what exocytosis looks like. Remember those vesicles that you created from your rough ER, and then it's repackaged in the Golgi body. And if this is a secretory proteins that you've produced and repackaged at the Golgi, then it's destined to be released out of the cell, right? So the vesicle will go to the edge of the cell, where it will then fuse with the cell membrane, and in doing so, it will be releasing its contents. Endocytosis is the exact opposite. If you have some particles on the outside that you want to bring into the cell, then there is going to be a folding that you form on the surface of the membrane, and then eventually this part here will merge with this, and it kind of pinches off from the cell membrane, and you create a small vesicle that will go into the cytoplasm. And if you want to break down the contents of the vesicle, of these green balls that you just ingested, then this will fuse with the lysosome, right? You remember from last lecture, then the lysosome is going to have these enzymes that will then break down the green circles here. Now, endocytosis actually has three different categories. The first one is what we call phagocytosis. Okay, phagocytosis means cell eating. So this is where the cell is going to be taking in large particles. And an example of that would be a white blood cell here engulfing some kind of bacteria. Okay, so it literally will ingest the whole thing, bring the whole bacteria in, and you might surround it with a vesicle and then break it down with lysosome. Okay, so that's cell eating, phagocytosis. The other one is penocytosis. Penocytosis means cell drinking, and in this form of endocytosis, you are basically bringing in mostly a fluid, okay, extracellular fluid. Okay, so there's no particles in it. It's just mostly water that you're taking in, and human eggs can actually take up nutrients present in the surrounding environment by doing penocytosis. And the last one is something called receptor-mediated endocytosis. So receptor-mediated endocytosis works very similar to phagocytosis, but then it's more picky about what the cell takes up. Okay, there are these special receptors that are located on the surface of the cell, and in order for you to be bringing certain types of material in, that material must fit into the receptor. Yeah, so cholesterol will be absorbed by the liver through this way. Here is a series of diagrams that shows you what receptor-mediated endocytosis look like. On the surface of the cell, you have these receptors that would fit these yellow circles. And what happens is, if you have some like triangle molecules here, and the triangle molecules bind to the receptors, they won't fit, and so you won't be bringing in the red triangles. So once the yellow circles bind to the receptors, that will cause the surrounding membrane to kind of cave in, and then eventually, like, this part is going to merge with this part, and you're going to pinch that off the surface of the cell membrane, and you will be able to take this in as a vesicle. So that's it, right? This is a final summary of all the different types that we've talked about. It's good to know what belongs to which category, and be able to give some examples of each type, and this is a written summary of all the types of transport that we talked about, and the requirements, as well as specific examples for that. So that's it for this lecture. Thank you for watching. If you have any questions, feel free to email me or ask me a question during the tutorial sections. Thank you for watching.